Hi there. In this video, we will look at different ways that people study cancers, emphasizing techniques which we've just been studying about. We'll look in turn at the way people analyze DNA in cancers, RNA in cancers, and proteins in cancers. And we'll end with a landmark study, again from the Weinberg lab, where a cancer cell was deliberately engineered from a regular cell. That might seem like an odd thing to want to do, but again, there was value in having a molecularly defined cancer rather than one that is the result of random mutation. Given the advances in sequencing technology, it's not surprising that one of the main ways people study DNA changes in cancer is through sequencing of tumor genomes. I've posted here 4 articles that were current about a year ago. I could easily put 400 on a slide today. The basic approach in all these studies is to sample a patient's tumor genome. Let's use the lung as an example. Cut out a little bit of the lung tumor, sequence the DNA that's present in that tumor, and then also take a very small piece of normal lung tissue some distance away from the tumor so that we have the patient's normal or unmutated genome and the cancer genome. You can also take the normal genome from a different organ, the blood, the cheek cells. There is variation in that element of the study design. But by sequencing both the tumor and the normal original genome sequence in a patient, one can identify mutations that are present in the tumor that were not present in the original genome. In all these cases, many thousands of changes are observed, and one of the trickiest aspects of this then becomes which of the mutations are causing the cancer and which are there without any effect. They happened and they're just going along for the ride. In fact, the terms for these two types of mutations are driver mutations or passenger mutations. And to summarize a large amount of data, what are some of the take-home messages? One of the big ones is there's only a very few proteins that are consistently found mutated in large proportion of the samples. If we're looking at lung cancer, only 4 genes are mutated in more than 20% of the cancers. 3 of these 4 you've heard a lot about: p53, RAS, and EGFR. The 4th protein was a bit of a surprise, STK11. That's a serine threonine kinase. It functions normally as a tumor suppressor. And prior to these studies, this gene was not perhaps recognized as a very prominent oncogene. But if we look beyond just 4 genes, we'll see that there are many, many genes that are mutated in a very small number of cancers. And this highlights one of the most confounding aspects of trying to develop therapies for cancers is that any kind of cancer, even lung cancers, even the most common type, non-small cell lung cancer, is a very, very heterogeneous disease. Mechanistically speaking, that's not surprising. Mutations arise stochastically. There's no reason to think that 2 people who both have non-small cell lung cancer would have exactly the same mutation profile. But I think expectations would have been that there might be a dozen or so genes that are mutated in large percentages, or that there would be sort of 3 or 4 main types of mutation patterns which will give rise to these various cancers. This heterogeneity points to an obvious problem. If all these lung cancers look the same, but they are in fact driven by very different mutation profiles, they might very well need to be treated very differently. It's seen time after time that a new attempt at a tumor therapy will be very effective in 5 to 10% of the population, but not effective at all, and in fact harmful. These drugs are poisons to 90% of the non-responders. This has certainly led to push to personalized medicine, where people's tumors would be sequenced and therapies tailored specifically for their mutation profile. And what we're learning to do now is to associate particular mutations with different treatment options. I will say that for all the complexity and the thousands to millions of mutations observed and the thousands of genes involved in the process, those decisions in terms of tailoring therapy right now rely on only a few proteins. is, for instance, EGFR mutated, then you're going to get erlotinib. Unless it has a second mutation, then erlotinib won't work. So for that case, the treatment decisions of those lung cancer cells will depend only on the mutation status of EGFR. You could just sequence that gene rather than the whole genome. But we sequence the whole genomes with the hope that we will learn information for the future to make better decisions. One way of reconciling the fact that most of these genes are not mutated in most cancers is by looking at pathways. And what is sensible and what is indeed found is that mutations might occur in multiple places in a pathway. This leads to the same phenotypic effect, but it is a reason why no individual gene has that high of a mutation prevalence in a cancer. If you think about the MAPK pathway, RAS, RAF, and then the three MAPK proteins, if any one of those 5 proteins gets stuck in a quote-unquote on position, you're going to have cell proliferation. It doesn't really matter. So you might a priori expect that of those 5 genes, none of them are mutated in more than 20% of the genes. So that is one trend in the analysis of the data one obtains by sequencing lots of tumor genomes. Another way that people study chromosomes of DNA, as you've seen with the karyotype pictures, is to look at regions with very high accuracy to see whether they're amplified or deleted. You'll remember the crazy colored mixed up pictures of the cancer karyotypes and sometimes multiple copies of a gene. And using DNA chips with different probes tiled in equal spacing along a chromosome. This is pointing out the location of the MYC gene that's on chromosome 8q because it's on the long arm of the chromosome. And so we'll take 2 samples, a cancer sample and a normal sample, or a pool of cancer samples and a pool of normal samples. We'll label one red, one green, and hybridize that, and we'll see where the ratio changes. If there are regions that are amplified in the cancer genomes, you would expect to find oncogenes in this region. If there are sections which are called tumor deletions, meaning that there are more copies in the normal chromosomes, and in the normal chromosomes there's only going to be 2, this is regions of the chromosomes where you might expect to find tumor suppressors. And here MYC, the dedifferentiation gene, is amplified in this particular class of cancers. So this is a technique called comparative genomic hybridization because we're putting it on chips. We're hybridizing genomes rather than RNA samples, which is what we've mostly used chips for and which we'll see more of in just a second. But here we're putting DNA on and applying it to a chip and looking for relative changes between the 2 sample types. The primary tool for studying RNA changes in cancer has been, up to this point, DNA microarrays. Either here is a spotted microarray or an Affymetrix chip. We'll take the 2 tissues, we'll isolate the RNAs, we'll label them different colors, we'll mix them, hybridize them to the chip, and we'll see genes that are upregulated in tumors with red spots, genes in green that are more highly regulated in normal tissue, spots that are yellow that are present in equal abundances. Now, this is a tool, a very useful tool for learning a lot about the biology of how things change when a normal cell becomes cancerous, but it's also been used diagnostically and prognostically. 2 papers came out back to back in 1999 that did similar things, and what they were doing is they were going to use microarray data to classify cancers. In this case, 2 particular types of leukemias, ALL and AML, which under the microscope look very similar. It's hard for pathologists to differentiate them. However, they respond very differently to treatment, so this is an important diagnostic criterion to make. What Todd Golub and co-workers at the Broad Institute was able to do is identify a set of genes that were upregulated in ALL, downregulated in AML, and a number of genes that had the inverse pattern. Again, genes are rows here, different patients or individual samples are the columns. And then, based on the gene expression pattern of these 50 or so genes, you are able to arrive at a conclusion on which type of tumor you are looking at and how you should go ahead and treat the patient. Now, there are a couple of interesting things when we look at this data a little more closely. The first off I'd like to point out is looking with your eye at these two patterns, you can instantly tell the differences between these two types of samples. That's something the human brain is pretty good at. Computers are less good at it. Furthermore, if we look at any particular gene, I'll just randomly choose one, but I'll leave it to you as a test to look at the other genes. No individual gene is going to correctly identify the presence of the tumors or not. As you can see here in the ALL, there are many blue spots, which is what they're taking as the signature of the AML spot. It's only when these genes are looked at in aggregate that if one is missing one of the high values, then one of the other genes will do, and it's combined in sort of a voting approach, is the way they described it, that you can arrive at a conclusion. Another thing that's worth noting is while some of these genes are ones that you might recognize, here's a cyclin, that makes sense that it's involved in a cancer. Many of these other genes, and in fact most of them, are not what's considered to be functionally related to cancer biology in any primary way. You can make a hand-waving argument for each of those genes, but a priori you would have not spent a lot of time talking about lysozyme and its role in these two leukemias as being a reason that you might expect a difference between these two samples. A final comment about this important data was that this was a case of supervised classification, meaning that the research team looked at a lot of data, selected these genes themselves to use as a classifier when looking at these types of tumors. In this study, again, coming out about the same time from Peru and co-workers, is they looked at different types of breast cancers. They did not do supervised classification, they did unsupervised classification. Once they had the expression data for lots of different breast cancer samples, they did hierarchical clustering on the sample types, and what they find is that there were several distinct patterns present in the breast cancers. So this is discovering subtypes of breast cancer, which, although it preceded the DNA sequencing data by a lot, is also supported by that, in that cancers are a heterogeneous disease and that there might be shared molecular defects that's causing the various patterns. Furthermore, this group went to use on nearly all of the data rather than selecting a few genes of particular import. That can make it harder to use in the clinic, but it's also perhaps less prone to bias that might interfere. The tumor subtypes they found also happen to have prognostic implications. Some of the types, such as the basal subtype of breast cancer, can have very poor prognoses. Some of the breast cancer subtypes, like luminal A, have very good prognoses. So this study was really landmark in showing that the systematic and comprehensive study of RNA molecules could not only identify subtypes, it could provide prognostic information about how a patient might fare. Now, prognostic information is not always the most helpful. If there are no good treatments, what does it really do for the physician and the patient to know you have a bad prognosis or you have a good prognosis, other than people like to know and you might get more aggressive in terms of treating these patients. And while there are a lot of prognostic studies, people are trying to identify subtypes that respond to different therapies. So people are classifying tumors as being drug X resistant or drug X sensitive. That provides actionable information for the physician and patient to make decisions about the healthcare. For the study of protein changes in cancer, I'll briefly mention some of our own work where we did mass spectroscopy on signaling pathways. We were looking at the phosphorylation status of the EGFR signaling pathway, a driver for lung cancers. We were very easily able to diagnose whether a phosphoproteomic pattern came from a normal lung cancer or lung cancer types based on which signaling proteins were phosphorylated or not. The code here, the color code, is similar to RNA legends. The pink or red colors are more phosphorylated in tumors. The green nodes are more phosphorylated in normal tissue. Yellow proteins were equally phosphorylated. Gray proteins, which are included on the basis of their inclusion in the signaling pathways, were not observed to be phosphorylated. And that was a limitation of the phosphoproteomics at the time. We know very well that retinoblastoma protein, for instance, is phosphorylated. However, it was not observed in our study. We've gone on to look at phosphorylation patterns to predict drug sensitivities and are encouraged by some of the results that we've obtained, again, in lung cancers. One of our motivations for pursuing this line of research relates to the problem of the thousands to many thousands of mutations found in DNA sequencing. It's not clear what the passenger mutations are at the DNA level or the driver mutations are at the level. In this case, we know that this is the pathway that is responsible for driving a large number of lung cancers. So we're looking specifically at the biological mechanism whereby lung cells proliferate. Conceptually, we feel that this provides the most pertinent information towards understanding tumor biology. Furthermore, not only is the biology determined by these pathways, but a huge class of new cancer drugs, tyrosine kinase inhibitors, specifically influence the phosphorylation status of tyrosine residues. So by using them to classify our tumors, we feel that we're looking at the molecular species that not only drives the biology, but is also the target of the treatment. Time will tell if this proves to be a fruitful approach or not. We'll end by looking at another landmark paper that came out in 1999, and this was the specific molecular creation of a tumor cell using the tools of recombinant DNA. Most cancer cells that occur are the product of random mutation, so we have heterogeneous behavior. And we don't even understand all the molecular changes that transformed a normal tissue into a cancerous tissue. These scientists took normal primary cells and introduced 3 new proteins and found that the resulting cells behaved exactly like tumors. The 3 proteins they included were telomerase, RAS, and a viral protein called SV40 large T oncoprotein. And let's look at what they might be doing. Now we know for cancers we have to have 6 properties that are going to be needed to have an invasive and metastatic cancer. These studies were done in vitro in petri dishes, so metastasis and invasion was not being assayed, but you still need to acquire 5 different capabilities in order to achieve the cancer's phenotype. By introducing telomerase, they have limitless replicative potential or immortalization. By putting in RAS, we're going to affect 3 of those. RAS is a protein that affects angiogenesis. It has an insensitivity to growth factors, and it also helps cells evade apoptosis. And I should say that they're specifically putting in the mutated form of RAS, the one that stays on no matter what. And then by putting in the SV40 large T antigen, this is a protein that's known to bind and inactivate 2 big players, p53 and the retinoblastoma protein. And so this is going to inactivate the cell's resistance to growth inhibition and will also lead to immortalization by evading apoptosis and continuing to allow cell cycle progression. So by entering 3 particular proteins, they are now able to convert normal cells into cancer cells. Okay, that's what I would like to say about study of cancer using genomic techniques. Thank you for listening.